Okay guys, so let's go ahead and start talking about the fourth part in the molecular biology of the gene lecture. This is definitely one of the longer lectures because there's just a lot of stuff going on here in regards to molecular biology. As I mentioned before, it's just such an exciting new field, and there's so many new developments occurring that content continues to grow and grow and grow as new discoveries are being made. Again, just quick re-emphasizing key points. We talked about Erwin Chargaff, his contribution, Rosalind Franklin's contribution, and then Watson and Crick, their contributions, the basic structure of the DNA molecule, how DNA replicates. And then in the last lecture, we were talking about that stuff called RNA. Again, remember, DNA is the code that says this is what you're going to express. That code gets turned into RNA. The RNA is just a kind of a shorthand version of the DNA. A little bit of a twist to it, single strand, slightly different sugar, and no thymine in the RNA strand. So then RNA gets organized, pieces are chopped out and removed. Those are called the introns. Those stay inside the nucleus. And right now, scientists say this is non-coding information. But honestly, we don't really know what it does. It could be far more important than what actually leaves the nucleus, what we call the exons, leaves the nucleus and goes and becomes an actual gene. So in the near future, I may be completely redoing my lecture to say, now we know what introns are, and this is what an intron does and how it works. Again, that's the fun part about biology, and especially molecular. It's changing all the time. Okay, so what we're looking at, and we talked about the intron staying inside the nucleus, the exons leaving. The exons are then going to go to the ribosome. And the ribosome is the key structure where this next step that we call translation occurs. So the messenger RNA is translated by the ribosome. Okay, so translation is the process of taking the RNA code and turning it into, there it is, translated, turning it into a string of amino acids or polypeptides. This happens specifically at the rough ER, more specifically at the ribosome. The ribosome is the structure that reads the messenger RNA information and dictates this will become this. It's always done in 3-letter codes we call codons. So 3 letters in the DNA alphabet become 3 letters in the RNA. 3 letters in the RNA, we call it a codon. That equals 1 amino acid. String together a bunch of amino acids to create a gene. On average, a gene is made up of 2,000 to 4,000 DNA letters. Okay, so just giving you an idea of rough amount of information it takes to create a physical expression of a trait. All right, so this next slide is a great slide. This is kind of the overview. Transcription happens inside the nucleus. DNA is the template, becomes messenger RNA. Introns are taken out and they stay in the nucleus, and then you have the mature messenger RNA. This strand leaves the nucleus, goes to the ribosome, and at the ribosome, this is where it's going to be read, or we say translated. So transcription's inside the nucleus, translation is out here at the ribosome. The ribosome then looks at the 3 letters, matches it up to an amino acid. There's your first amino acid. Next group of 3, second amino acid. Next group of 3, third amino acid. Next group of 3, fourth amino acid. So on and so on and so on, creating a protein structure. Now that structure gets packaged, processed, put into a little bag, and then shipped. So there's your Golgi body, or your UPS structure. Now it comes into the picture and says, hey, let me move that protein outside the cell or over to the cell membrane or wherever. Golgi gets involved. But it's got to be produced and formed at the ribosome. All right, so there's our overview. And like I said, that slide's a great overview of the two processes or two steps when we talk about gene expression. Now, sometimes mistakes occur. We call these mutations. It's a change. All right, so here's the DNA up top here. This is the DNA information. That DNA is supposed to turn into RNA, or right here we'd say this is DNA being transcribed into RNA. The RNA then is down here, we'll call it step 2, translated. So step 1, step 2. DNA gets transcribed into RNA. RNA gets translated into an amino acid. Tyrosine is an amino acid. So there's your normal code. Sometimes it's only one letter in the alphabet that gets messed up and everything changes. So if you come back here and you go, okay, ATG is your normal code. Now we have ATA. There's your mutation. But if you notice, it still codes for tyrosine. So it's a non-functional or non-influencing mutation. Nothing changes. You still get tyrosine. But if you go ATC in the DNA strand, now you have UAG, and we actually have a stop codon, or a stop protein. Uh-oh. So now your protein chain is done. Instead of maybe 7 amino acids, maybe it's only 3. That screws it up. Change the shape, change the function. Or maybe you mess something up here, and you change the A into a G. That G gets transcribed into a C. That gets translated into the amino acid histidine. So now you have a completely different amino acid there. Instead of tyrosine, you have histidine. Okay, that's a problem again. Those are examples of mutations, changes to the strand. So a real-life example is sickle cell anemia. Our normal red blood cells over here are nice, they look kind of like jelly donuts. But if somebody inherits the genetics where this amino acid, glutamate, is, instead of being expressed as glutamate, is expressed as valine, now you have sickle cell anemia. And your blood cells have a different shape. They are not functional as normal red blood cells. They have that curved kind of sickle shape here, and it doesn't carry oxygen. It leads to a lot of health problems simply because of a change in the DNA code. So here's how easy it is to get the code changed or to express a different amino acid. Okay, so normally, let me back up. Normally we want glutamate here. So glutamate, normal, I'll do normal in green. Normal coding for glutamate is either GAA or GAG. So if we come down here, GAA, GAG. Those are the two codons that will give us glutamate in our amino acid chain. But if something happens and you inherit this mutation from mom or dad, or mom and dad, then instead of GAA, maybe you have GUC, or you have GUA. I mean, that's one letter difference. Instead of an A, you have a U in the center there. Or GUG. So instead of an A in the center, you have a U in the center. That gives us valine instead. So valine, GUC, GUA, GUG. 3 ways to get valine. Any of those is at that particular amino acid point is going to lead to sickle cell anemia. The protein shape changes, so that causes the shape of the red blood cell to change. And if you bend that red blood cell into that sickle shape, so instead of our normal kind of jelly donut, it kind of curves into this, that sickle shape, it can't carry oxygen as well. Lack of oxygen leads to a whole laundry list of health issues because of low oxygen levels. All right, so key things to remember here when we're talking about DNA and gene expression or DNA turning into proteins. Mutations can lead to new genotypes, new pieces of DNA. New genotypes can lead to new phenotypes, new expressions of traits. So now you have this new trait being expressed. A new phenotype can be good or beneficial. It could be neutral and really doesn't make a difference, or it could be harmful. It all depends upon, and let me do this in bold, depends upon the environment that the phenotype is expressed in. So if it's expressed in a certain environment, that new physical characteristic may be incredibly beneficial, and the organism survives and increases its chances of reproductive success, passes on its genetics, more individuals inherit the trait, and it's a positive thing. Or it could be neutral. Again, no big deal. Nothing happens to it. Or it could be very harmful, and the organism doesn't survive, and it's not likely to pass on that trait, and the individual dies. All right, this is one of the major driving forces behind evolution. So we're going to talk about evolution in further or future lectures. Evolution is the change in the genetics of a population. Mutations are how genes change. Good mutations benefit the organism, tend to increase in the number of individuals who have it, and they're positive, and all of a sudden more and more and more are showing up with this particular trait because there's an advantage to having it. Bad mutations, again, tend to kill the organism. They tend to weed themselves out of the population. But neutral mutations can be retained in populations as long as they're not harmful. So everybody who has blue eyes, that's a mutation that showed up in a common ancestor from a northern European environment. And that mutation did not show up until about 6,000 to 10,000 years ago. So consider human population. People who have a very strong and fairly recent ancestry. And we're talking 4, 5, 8, 10, 20 generations, hundreds, thousands of years ago. If your ancestors were in Africa, the Middle East, Asia, etc., they all had dark eyes. It's the European ancestors that were around 6,000 to 10,000 years ago that had the blue eyes or lighter eye color, and that got passed on through the generations. Light eyes in Europe, Northern Europe, didn't hurt at all. But take light eyes or blue eyes to Africa, it's hard to see. More solar exposure is harsh and difficult on people with light-colored eyes, but people with dark-colored eyes in Africa, sun exposure doesn't hurt as much. It's not as difficult on your eyes if you have dark eyes and you're in an environment that has a lot of sun. So think about that. You look at somebody's eye color, you can get a very general picture of where their ancestry is from. You have no guarantees, but if you have blue eyes, your ancestors 6,000 to 10,000 years ago were sitting in Northern Europe. If you have dark eyes, odds are your ancestors were in one of those other environments. Now, our poor frog over here, that's just a horrible mutation. Back legs are deformed, not developing correctly. That frog's not going to survive. It's not going to develop. It's not going to be able to reproduce, and it's going to get weeded out of the gene pool. Genotypes and traits only survive, again, if they're advantageous or beneficial. They increase the fitness, the ability of the individual to reproduce, or if they're neutral and they don't cause any harm. But harmful ones, again, tend to get weeded out because, you know what? If it hurts you and you can't reproduce, you can't pass it on to your kids. All right, so a lot of really cool stuff going on with molecular biology and biotechnology. So keep in mind this field is constantly changing.